Hello, grade 11 students. I hope that you're doing good and that you're safe. So welcome to this video. I will do a quick overview about what we covered previously and also about mitosis. Okay, so first of all, we said that the DNA is the part that carries the genetic information from parents to offspring. Okay, this genetic information is located in the nucleus of cells. When the DNA is very coiled around proteins, histones and non-histones, we say that the DNA is condensed. And then chromosomes are well visible and we can clearly see their X shape inside the nucleus. Okay, cells can be diploid. This means that they have the whole set of chromosomes, okay, the set coming from the mother and the set coming from the father, or haploid, okay? When the sex cells, so during fertilization, when the sex cells meet, when the sperm cell fertilizes an ovum, so it's a haploid cell and another haploid cell, we obtain the zygote, which is diploid, which is the first cell of an embryo, okay? We need to get a multicellular organism, a full organism, out of this one cell. So what to do? The cell has to undergo division. Okay, so the cell undergoes mitosis. Mitosis is the process by which a cell, a diploid cell mainly, okay, a cell of the body, we're taking here the example of a zygote turning into an embryo. Okay, so it is the process of making same copies of the mother cell. By the end of each mitosis, we obtain two identical daughter cells. These cells are also identical to the mother cell, and this way the genetic information is conserved. Okay, why do we need mitosis? For several cases. The first one, as I told you, the zygote is the first cell of an embryo. So in order to develop, to make an organism out of this one cell, this zygote undergoes mitosis, okay? Another example is to renew or replace injured or dead old cells. Okay, remember that mitosis, so this yellow part right here, is just a part of a cell's life cycle. So what is the other part? The whole other part is called interphase. Interphase consists of three phases: gap 1, synthesis phase, and gap 2. Okay, interphase is not a part of mitosis, but to prepare for mitosis, a cell must undergo interphase. Okay, what happens during interphase? Let's see. During the first phase of interphase, which is G1, okay, so right here, it's the first checkpoint. The cell duplicates all the organelles that are present inside the cell. During the S phase, which is the synthesis phase, we duplicate the DNA. As I told you, we started mitosis with a diploid cell. So the cell has its full set of chromosomes. So 2n in humans is equal to 46 chromosomes. We do not double the number of chromosomes. No, we double or replicate the chromatids. So instead of having 46 chromosomes with one chromatid, we will have 46 chromosomes with two chromatids. Okay, this is very important since we need to conserve the number of chromosomes at the end of mitosis. After the S phase, we have the G2 phase, which is the phase of preparation for mitosis. Okay, once mitosis begins, G2 ends. So during G1, organelles are duplicated. During S phase, DNA is duplicated. During G2, this is preparation for mitosis. What happens during mitosis? Mitosis consists of 4 stages, 4 steps. You can memorize this acronym, PMAT. P for prophase, M for metaphase, A for anaphase, and telophase. During prophase, many changes happen in the cell. Chromatin condenses. This is why we will see visible chromosomes. They will be individually distinguished, يعني visible. We will see clearly their X shape. The membrane of the nucleus disappear. We will see the spindle fiber forming. These are the spindle fiber, okay? So this is early prophase. As you can see, the spindle fiber are forming. This is the start, this is the beginning. And also centrosomes, which are special organelles, they change into asters. So now we have the asters. This is early prophase, since as you can see, the membrane of the nucleus is still present. At the end of prophase, okay, the membrane of the nucleus is not present, and the asters, look, they are at the 2 opposite poles of the cell. So chromatin condenses, chromosomes are now visible. We can clearly see their X shape. The membrane of the nucleus disappears. The spindle fibers appear, and the centrosomes form into asters, and then they move to the 2 opposite sides of the cells. Then we have metaphase. During metaphase, the chromosomes line up in the middle of the cell. They form the metaphase equator, or we call it equatorial plate. So during metaphase, chromosomes line up in the middle of the cell, forming the equatorial plate. Now, what happens? Look, they have their X shape. So they have 2 chromatids. These chromatids were separate during anaphase. So we call this polar ascension. It's when the 2 chromatids separate and each chromatid moves to a pole, okay? So each chromatid moves to the opposite pole of the cell. This process is called polar ascension. Polar ascension, يعني the chromatids are separating and moving toward the poles, the 2 opposite poles of the cells. Till now, everything is happening in one cell. We still didn't divide the cell. So prophase, metaphase, anaphase. Now the last phase or step in mitosis is telophase. What happens in telophase is exactly the opposite of what happened during prophase. Okay? Look at these chromatids. They are on the opposite poles of the cell. Now, the membrane of the nucleus, we lost the membrane of the nucleus during prophase. It disappeared. هلا it will reappear. So the nuclear membrane reforms, so it reappears. The spindle fibers disappears. Asters, they return to the centrosome state. They will come back to the centrosome state. Okay? Chromosomes, they decondense. So during prophase, the chromatin condenses and chromosomes become visible. Here the opposite. Chromosomes decondense. Then, chromatin are back. To split these 2 cells, you will see a cleavage furrow at the center of the cell. We call this a cleavage furrow. to split the mother cell into two identical daughter cells. Okay? This splitting process is called cytokinesis. So by cytokinesis and through the formation of a cleavage furrow at the center of the mother cell, we divide the mother cell into two identical daughter cells. Okay? During mitosis and at all stages, يعني prophase, metaphase, anaphase, or even interphase, the number of chromosomes is 2n. What changes is the number of chromatids. Okay? يعني during interphase, بالG1, we had 2n chromosomes but with one chromatid. During the synthesis phase, we duplicated the chromatids. صار عندي 2 chromatids. G2 we did nothing. بعد عندي 2n chromosomes with 2 chromatids. Okay? Prophase, 2n chromosomes, 2 chromatids. During metaphase, the chromosomes are lined up on the equatorial plate, 2 chromatids. Okay? During anaphase, we are still in the same cell. We still didn't split the cells. 2n chromosomes, 2 chromatids. All the chromatids are present in the same cell. Telophase, splitting, cytokinesis happened. يعني we have 2 daughter cells. Each one of them have the same number of chromosomes. بس هلا the chromosomes have 1 chromatid. As you can see, look, at all stages we have 2n chromosomes. 2n chromosomes. The number of chromosomes is conserved. It doesn't change. This is why I told you at the beginning, mitosis is a process to obtain daughter cells that are identical to each other وكمان identical to their mother cell. They have the same set of genetic information. Okay? There is a little bit difference between plant cells mitosis and animal cells mitosis is that in plant cells there are no asters and no cleavage furrow. Okay? هون بالanimal cells, we can clearly see that there is a cleavage furrow splitting the cell into 2 daughter cells. Okay? بس بالplant cell, this does not happen. بس a cell plate appears at the middle and split the mother cell into 2 daughter cells. Okay? This is all about mitosis. In the upcoming videos, I'll be talking about the second type of cell divisions, which is meiosis. It starts with a diploid cell, but it ends with haploid cells. Okay? So take good care, and I hope to see you soon.